Experiment. My name is Marty and I'll be teaching you about what we are studying today.
Hi, I'm Dale. I will be explaining the experiment for you.
My name is Asani. I'll be breaking down our data and analyzing phase for today.
Hi, my name is Sally. I'll teach you how we plan our experiment.
Hi, I'm Matthew. Today I'll be lecturing about our experimental organism to you.
Recently, we students learned about circadian rhythm in our science class. To give us a visual of circadian rhythm, we performed an experiment using an organism named Pyrocystis fusiformis. The organism has a circadian rhythm that is very easy to observe. I just learned that, coincidentally, Pyrocystis fusiformis in large groups uses a lot of carbon dioxide. We are now studying the carbon cycle. But anyway, I'm going to turn it over to Matthew to teach you about Pyrocystis fusiformis.
Oh! Hi everyone. I was just practicing my song for the Unicellular Protist Music Festival. Can't wait for it. I hear even Zua Zandali, at least that's what Hassani said it was called, will be there. So excited. But never mind that. Allow me to explain one of the most unique creatures in the ocean to you. Pyrocystis fusiformis. Pyrocystis fusiformis is a flagellate protist. It is a marine plankton that is photosynthetic. Feel free to take a look at your screen and follow along as I classify Pyrocystis fusiformis. We will use the six-kingdom structure for this classification. The domain of Pyrocystis fusiformis is eukaryota. Eukaryota organisms have cells that contain complex structures and close within membranes. The defining characteristic of eukaryota, though, is that every organism with a nucleus or nuclear envelope is a eukaryota. Although the kingdom of Pyrocystis fusiformis is up for debate, we are today going to classify it as a chromoviolata. The debate is if chromoviolata even is a kingdom, but that doesn't matter for us. Chromoviolata organisms have a shared origin of chloroplasts, and there is presence of cellulose in most cell walls. Pyrocystis fusiformis is classified under the superphylum of Alveolata. Organisms in the superphylum of Alveolata are close relatives based on ultrastructural and genetic similarities. Alveolata is the main line of protists. Pyrocystis fusiformis is a dinoflagellate, and Dinoflagellata is a phylum. Dinoflagellates are unicellular organisms with two flagella. They have a complex cell covering called amphiesma, chloroplast bound by as much as 3 membranes, and fibrous extrusum. A dinoflagellate has a rough or smooth endoplasmic reticulum, Golgi apparatus, mitochondria, lipid and starch grains, and food vacuoles. The superclass Dinocaryota, which Pyrocystis fusiformis is a resident of, is a dinoflagellate that has a nucleus that remains a dinocaryon throughout the entire cell cycle. Little more information is widely available in regards to the scientific classification of Pyrocystis fusiformis. Pyrocystis fusiformis falls under the order of Gonualcales, the suborder of Goniodominae, the family of Pyrocystaceae, and the genus of Pyrocystis. The species, though, we were able to find quite a little bit of information on. Pyrocystis fusiformis is a marine plankton with the ability to produce bioluminescence in response to water movement and high shear flow. Pyrocystis fusiformis can produce up to 62 light flashes every second. Each flash lasts about 210 milliseconds. These flashes have a photon intensity of up to 690 by 109 photons per second. The bioluminescence is stimulated by shear flow, velocity gradient, or low pH in the 5 to 8 range. The average organism is approximately 970 by 163 micrometers.
Wow, that was long. Now I understand why he said he was going to lecture us. Anyhow, the light flashes made by Pyrocystis fusiformis can only be seen in the dark. Like at night, the organism performs photosynthesis and chemical energy reformatted from light energy. Once again, becomes light energy during the flashes.
How does the color of the vial containing Pyrocystis fusiformis affect its bioluminescence?
Bioluminescence is a key term here. We were very curious about the color in all this. We knew that sunlight was red, orange, yellow, green, blue, indigo, and violet. We also knew that the Pyrocystis fusiformis light was blue. So we thought that if we interfered with sunlight by placing colored film around the Pyrocystis fusiformis samples, it would alter their photosynthesis, or watercolor. Or place Pyrocystis fusiformis go? Would it maybe 2 or all 3? We then knew what to watch for. We had our question, the first step of the scientific method.
Our group was split during the second step of the scientific method, hypothesize. We were not able to find a compromise between the two predictions, so I went ahead and wrote down both. Just so you know, we chose to wrap some of the samples in blue cellophane.
We predict that when the vial is coated in blue cellophane, the organisms will give off more light and more intense light. We came to the conclusion because of the sleep article we read earlier this year.
In the sleep article, it said that when living things get blue light, they are more alert.
We think that when there is cellophane wrapped around the vial, it will make almost all the sunlight into food, so at night there will be more alert and will light up.
We predict that the Pyrocystis fusiformis organism in the experimental group will not be able to function as well as the Pyrocystis fusiformis in the control group. We believe that when radiated light makes contact with the blue cellophane wrapped around the vial of the Pyrocystis fusiformis, the only light that passes through onto the Pyrocystis fusiformis will be blue.
Recently, we read an article that explains a scientific study relating to blue light. The conclusion the researcher came to is that blue light keeps organisms awake. We hypothesized that since only blue light will contact Pyrocystis fusiformis, the organism will be constantly awake. This leads us to theorize that the loss of sleep, resulting in less energy, will prevent the experimental Pyrocystis fusiformis from ideal daily function, including photosynthesis to an ideal ability and lowering the amount of chemical energy that Pyrocystis fusiformis can use to perform its bioluminescent routines.
Our experimental variable was if there was blue film on the vial of the Pyrocystis fusiformis or not. We tried our best to keep everything we could control, like where the vial should be kept, when the data should be collected, and so on and so forth. Here's a look at the table we used to collect data during the most imperative part of the scientific method, step 3 experiment.
Here are the results for our experiment. As you can see, Dale and I are not in the table. My Pyrocystis fusiformis died the day I died, and Dale's ended up across the city. By the time he got to it, it was dead. So Marty ended up being the only control group. Her Pyrocystis fusiformis vial lit up completely for more than half of the experiment. Then we assumed that it must have died. Matthew and Sally both had blue cellophane on their vials, and their data was everywhere. Sally's became fairly consistent near the end of the experiment, but Matthew had data at both ends of the earth. The line graph should help you picture the data table a little better. Our conclusion is that based off of our observations, we cannot conclude the answers to the question we asked, which was, how does the color of the vial containing the organism, Pyrocystis fusiformis, affect its bioluminescence? We agree that our experimental groups were affected by the altered light. However, we also agree that we cannot settle on how precisely or why it is true. Since the question data we collected was very different from each other, we decided that we cannot answer the question.
We believe that no matter if the method used during the experiment is excellent or terrible, it should be analyzed by the people who use it. We think the things we could have done better or have been better prepared for our experiment, more detailed in our experiment method, and have tightened our controls to make the experiment as controlled and accurate as ideally wanted. If we had been able to do this, we would have liked to have larger sample size in case of the dead organism, and for the most accurate results overall. Moving forward on our work, questions we have to build off of this experiment are:
how would the intensity of the color of the vial affect the Pyrocystis fusiformis?
How would the type of lights, such as LEDs, fluorescent lights, and natural sunlight, affect the Pyrocystis fusiformis?
How would the intensity of the light contacting the organism affect Pyrocystis fusiformis?
And how would the color of the vial containing the organism affect species scientifically related to Pyrocystis fusiformis? Thank you for watching.
Hi, my name is Bob, and I approve this message.
I got the magic in